This is a quick review for the first test. Sorry we've been having so many complications with this test. Yeah, some of you weren't able to watch the videos. Those links are now gone. We're downloading new links so that you can watch them directly from Moodle. You don't have to be kicked out to YouTube to watch it. So I'm hoping that by Wednesday morning, all those videos will be there and set and ready to go for chapters 1 through 4. I've also posted chapters 5 through 7 for the second test, but we'll talk about that later. Right now, the test will be Friday morning from 10 in the morning till 11 at night. And once you log on a Proctor Track, you have 90 minutes to complete the test. And so once you log off and you log back on, that you can't log back on. So you get this one shot at it. Don't take any breaks during it. Proctor Track's going to watch you and record what's going on while you're taking the test. So so be ready Friday morning from 10 to 11 at night. It'll probably only take you about 40 minutes to take the test, but I'm giving you 90 minutes just to make sure that everybody can go at their own pace and go slow. You can go back, once you go through the test, you can go back and review them again. You can change answers if you want to. But once you save and enter the test, that's it. You can't get back into it. So, and it will let you know after you take it the points, but just realize don't go by the points. Don't go by the grade in the grade book in Moodle. I will probably be curving these tests some, so don't freak out if you didn't do that good. I will usually curve them up sometimes up to 10 points. So we'll just have to wait and see what these tests look like. I try to make it as easy as possible. So let's go through this real quick. We're going to go through 4 chapters. Real quick here, and I'm just going to highlight to you some of the things that you really need to focus on so you can do well in this test. All right, know this: nature is everything in the universe except what humans have manufactured. That's a great thing to know for the test. And on the test, be able to know the organization of matter and life on this earth from the smallest part to the largest. So atom, molecule, cells, organism, tissue, organ, and then organ system, then population, community, ecosystem, biosphere. Just know that general organization, from the atom being the smallest unit of matter up to the cell, the smallest unit of life, and we go on from there. All right, let's get through all of these real quick. Say we're going to go through these fast because you already have all this for your lectures. All right. Know that there's 3 things that we talk about that defines life. Know these 3 things. They need a constant input of energy and raw materials. They sense and respond to change, and they pass DNA off to their offspring. So know those 3 basic things for this test. Know that energy flows one way through the ecosystem. It can't be recycled. And as it goes from level to level, most of us lost this heat. So that's that second law of entropy, which we'll get to after a while. And nutrients, though, can be recycled. So living things, know these 2 general things. Producers make their own energy, make their own food. Consumers have to get it somewhere else. So know what a producer, know what a consumer is. All right. Know this term, homeostasis. Know what homeostasis is. Homeo means the same, stasis means standing. Your body or different systems are going to fight to keep the same standing. The amount of oxygen in the blood, the pH of the blood, the amount of sugar in the blood. All these different things is an example of homeostasis, body temperature. All right. They sense and respond to change, and they use DNA. All right. Know that there's 2 basic divisions of life. Remember, they have a nucleus or they don't have a nucleus. So the eukaryotes and the prokaryotes. Prokaryotes do not have a nucleus, eukaryotes do. So bacteria and archaea are the eukaryotes. That's two domains that we're looking at. No nucleus in bacteria or in archaea. So there's bacteria, archaea. But here's our three domains. Know the three domains of biology. You have bacteria and archaea, no nucleus. Then everything else that has a nucleus is the eukaryotes, the eukarya. And that's everything from the protist to the fungi, to the plants, to the animals, all have nucleus. So there's our eukaryotes. Know the definition of that. Know what a protist is? A protist is the simplest, it's a whole kingdom. It's the simplest organisms that have a nucleus. So it's everything from single-celled organisms to giant kelp. And just examples. Know the definition of fungi, and remember what I pointed out to you. Look for secrete substances that break down food outside of the body. That's the giveaway for fungi. They decompose dead things outside. They secrete their digestive enzymes outside the body. It digests, and then they absorb it back in. Examples of fungi. Know a quick definition of plants. That should be easy. They're producers. They have chloroplasts. They make their own food through photosynthesis. Animals, just a basic definition of what an animal is. All right, so let's see here. What do we need to know here? Know the definition of what a species is. They're based on shared traits, but also they can reproduce and have viable offspring. That's the basis in the animal and plant kingdom and the fungi and the protist of what a species is. Know the difference between a genus and a species. Remember, it's that binomial system. Know Carlos Linnaeus, he came up with the binomial system, a genus name and a species name. And so remember, we talked about humans. We are Homo, genus, sapiens, species. And so the genus is always capitalized, the species is not, and the genus species name is either italicized or it's underlined. So the example, Rosa canina for the dog rose. All right. Here's our three domains again. Know those three domains. And there's the six kingdoms. I'm not going to ask you the six kingdoms. Know the three domains. The six kingdoms aren't important right now. We'll get to that later. All right. Some of this stuff we're just going to go through pretty quick. Here's the species concept. Here's where we get that individuals that potentially can interbreed and produce fertile offspring. So that is what a good definition of species in everything but the bacteria and archaeans. Know what natural selection is? It's where the strongest genes are passed on. The strongest genes survive. It's survival of the fittest is what Darwin called it. DNA is the basis for our information. Know for the test, and it'll come up when we get to nucleic acids too, but what is the only thing that DNA codes for? What's it got the recipe for? It's the blueprint to build what? And it's proteins. Know that for the test. Know a general definition of evolution, natural selection. That's a good thing to know. All right, scientific method. I'm not going to ask you individual tests like we talked about, this one for Alestra with Procter & Gamble. Know that you start with a hypothesis, you make predictions, you conduct your experiment, and you come to a conclusion with it. All right. Know basically what sampling error is. The larger the number that you can use in your test group, the better off your results are. If you just use one or two, then you can't trust your results. Talked about that. And know this. What's a scientific theory? It's a hypothesis that has been tested again and again and again, and it's have not been proven false, and so it becomes a theory. All right, so let me go now to chapter 5 PowerPoints. I'm sorry, chapter 2. I'm already thinking of the next test. All right, chapter 2 is about the basis of life, chemistry and life. Talk about, oh yeah, I got the speakers on. Mute that. A friend of mine got into my PowerPoints and added sound to it, which was these hideous laughs and different things. All right, so chemistry. All right, what is matter? Give me a basic definition of what matter is good. Know the atom is the smallest particle of matter. What's the smallest unit of life? The cell of matter is the atom. Atoms have protons and neutrons and the nucleus and electrons that circle it. Atom with a neutral charge has equal protons and electrons. Remember, protons are positive, electrons are negative. What is the atomic number? Know that for the test. That's the number of protons in the nucleus. Protons only. That's the atomic number. Atoms, electrons, there's our talking about charge. There's the nucleus, protons, neutrons in the nucleus. All right, what is an element? It's any substance that just has one type of atom, so that would be a decent thing to know in the test. Know this guy, Dmitri Mendeleev. He developed a periodic table of elements. A genius guy. Russian, a hermit. There's a picture of him. But man, this periodic table, he was dead right on. There's been no changes made to it. A brilliant guy. I'm not going to ask you really about radioisotopes. Don't worry about any of that. Know what the mass number is. It's the number of protons and neutrons. The atomic number is protons only. Mass number, protons and neutrons. All right. Don't worry about that tracers, radioactive decay. All right, know the basics of the shell theory of electrons, that shell model, that an atom is happy as long as its outer shell is full. So know the basics of that, which will come into play as we talk about other things. A molecule is the smallest part of a compound that has the properties of that compound consists of one or more atoms bonded covalently. So that's a molecule. All right, know what an ion is. It has either a positive or a negative charge, either because it lost an electron, it fill up its outer shell, so now it's positive. If it gains an electron, it becomes negative. And that electronegativity is the ability of an atom to pull electrons out. All right, what do we need to know here? Let's keep going. Still repeating. Know what an ionic bond is. Oppositely charged ions are attracted to each other. Remember we talked about this with sodium and chloride. He just needs to get one. If he gets rid of that, his outer shell's full. This shell's gone. He has 7 in his outer shell. He can fit 8. He needs one to be full. So very easily, sodium will give an electron to chlorine, and so now he becomes positive, he becomes negative, and since they're right there together because they just swapped this electron, they naturally will come and stick together because opposites attract. Like, the ends of the north and the south pole of a magnet will come together and stick, and that's what they do. So that is an ionic bond. Know what a covalent bond is. That's sharing electrons. Now sometimes, remember, one atom will pull those electrons a little bit closer, so that causes a polarity, a slight charge in these covalent bonds, but it is sharing. So nonpolar is when they share equally. Doesn't happen very often. Polar covalent bonds, one pulls it a little bit closer to it than the other, so it's slightly charged. Know the life-giving properties of water. That's going to be a good thing to know. Water molecule is slightly polar. and it forms hydrogen bonds. You know what a hydrogen bond is. So there's the definition of a hydrogen bond. Know that for the test. That's a good thing to know. So water is essential for life. Why? Because of all the hydrogen bondings. It has no charge. And whatever you need to remember, they break apart and reassemble so easily. Whenever it freezes, they're not packed as densely as when it is liquid, so it floats, and then remember that acts as an insulation for the water underneath, so the whole water column doesn't freeze, just that layer on top, and then insulates, because water can only go down to 32 degrees, so you got this barrier between the really cold air up top and the cold water, not as cold water below. Know what a solvent is, and know that water is a great solvent. Know that when water evaporates, it cools down the surface underneath it. That's why that's important for life, so we can maintain our body temperatures. Remember cohesion, that's going to be really important for water. I love that picture there. pH is critical too. Water can quickly change pH, but it's also can be buffered really easy. All right, so that is chapter 2. Let's go into chapter 3 real quick. Chapter 3. All right. Talked about our organic molecules are made up of carbon, with a carbon backbone. And the 4 elements of life, 4 molecules of life, carbohydrates, lipids, proteins, and nucleic acids. Don't worry about I'm not going to ask you about the models. Know what a monomer is? That's a subunit. Remember, as we talk about the example of carbohydrates, the monomer of carbohydrates is a monosaccharide, a simple sugar. But then those simple sugars can be put together, and that forms an oligosaccharide, like sucrose. But then more can be put on, and it forms a big, giant molecule, a polysaccharide, starch or glycogen, things like that. Know what condensation and hydrolysis is. Know the difference between those. Know those definitions for the test. Remember, I'm not going to ask you to functional groups, so be thankful for that. You don't have to memorize all of these functional groups. All right. Know what reactants and products are in a reaction. So carbohydrates are the most plentiful biological molecule in the biosphere. Cells use carbohydrates to build things. They use it for energy, and it can be stored as for carbohydrates in use. Plants store excess sugar as starch. So, all right. There's monosaccharides, oligosaccharides, polysaccharides. Another difference between those 3. Here's just a reaction to show you an oligosaccharide, a glucose and a fructose added together forms a sucrose. So let's go back and see, just so didn't know it was going to be that far back. All right, condensation. 2 small put together to form 1 large. So glucose, fructose joined together in condensation forms a sucrose with water added. So that was just an example of that real quick. All right, there's where we were. Know what a polysaccharide is. Know what cellulose is in plants, and know what starch is in plants, and know what glycogen is in animals. It's how sugars are stored. Cellulose is how sugars are arranged together to form that structural, to build up the body of the plant. Here's a starch where it's just that would be used for energy. Here is glycogen in humans. Each of these is a sugar put together to store energy, and then we will turn that into a fat with the fatty acid tails. There's cellulose, starch, glycogen, All right, lipids. Know what a lipid is? Know the difference between a saturated fat and an unsaturated fat. Know that on the test. I'm not going to ask you triglycerides. All right, know that wax is an example of a lipid. Steroids are examples of a lipid, such as cholesterol. All right, so proteins. Know that proteins, the shape equals the function. But what's the monomer of a protein? Amino acids. So the structure of amino acids, there's 20 different building blocks of those. 9 are essential for us to be getting on our diet all the time. Remember, there's 3 different structures. Remember, for proteins, shape means everything. Once a protein loses its shape, it can't do the job it was made to do. So there's the primary structure, the sequence of amino acids, the secondary structure as it is folded or coiled, and then the tertiary is where it is folded again to give it its active site. And there's a quaternary structure, which is if there's more than one protein added together to build a larger molecule. All right, so that's just all of that. Know that when a protein loses its shape, it's said to be denatured, and what can cause that? Heat, changes in pH, salt levels, and detergents can break up those hydrogen bonds that keep protein in its 3-dimensional shape. So when it loses its shape, it is denatured. All right, let's see here. I could ask this, depends on which example of the test I use. Sometimes I ask prions, it's just a misfolded protein. And so that things can't function, and so, mad cow disease, Creutzfeldt-Jakob disease are examples of that. All right, nucleic acids, know that they're the information. That's our DNA, RNA, ATP. What is the monomer? It's nucleotides or nucleic acids. So ATP is an example. Remember, that's that energy unit of the cell. And there is nucleic acids. RNA is an example of a nucleic acid. ATP functions as an energy carrier. DNA, which is decoding to make protein. So what is the only thing that proteins code for? I'm sorry, what's the only thing that DNA codes for? It's proteins. Know that for the test. That's it. Your DNA is how to make different proteins. But proteins are all important as they heavily function in the body. All right. It says the production of RNA and proteins. Realize RNA is just to make a copy of your DNA so that that can go to the ribosome to make the protein. So it's just a copy of your DNA to make the proteins outside of the nucleus. All right, just a great little slide to help you study that chapter. Okay, so that was chapter 3. Just one more chapter to go for the test. Chapter 4. All right, know that the cell is the smallest unit of life. I can guarantee you that's going to be on the test. What is the smallest unit of life? Pick a cell. Know the 3 things that all cells have in common. Know that. There's a lot of questions on the test from this chapter. Here's the 3. Plasma membrane, cytoplasm, DNA. All cells have those 3 things in common. Know that. Know that cells are small. I like these guys. I like asking you guys on tests. So far we have Dmitry Mendeleev, who made the periodic table of elements. No, Antoni von Leeuwenhoek, who was the first to describe microscopic organisms, that life could be small. He called them animalcules and beasties. No, Robert Hooke, he was the first to sketch and name cells. Brown was the first to identify the nucleus of a cell. So know these guys and their contributions to science. There's von Leeuwenhoek. Know the cell theory. Know the 4 parts of the cell theory. and know who proposed them. In 1839, Schleiden and Schwann, I can't come up with better names than that. Schleiden and Schwann came up with the cell theory. You know that. So, again, there's the three things that all cells have in common: plasma membrane, cytoplasm, DNA. You know that. Here's a eukaryotic cell, has a nucleus. Prokaryotic cell does not have a nucleus. That's a repeat. But that's great. We love it when things repeat. There's not as much to learn. Know what an organelle is inside of the cell. All right. Why are cells small? What's the main thing that keeps them small? Know that it is surface to volume ratio. I'm trying to hurry through this. All right. That's the main thing that keeps cells small, because plasma membranes can only hold in so much, and as the cell gets bigger, it puts exponentially more pressure on the outer membrane. All right, cells are very small. We don't need to, I'm not going to ask you anything on the test about that. Everybody knows cells are small. There's a prokaryotic cell. Know what a flagellum is. Know what a pilus is. Know what the capsule is. Usually I ask that more in lab, but we're not doing lab this summer term. So there's a definition of those. Know what a ribosome is. Ribosomes are in every cell. You know, there's three things all cells have in common. Could really be four. Ribosomes is where proteins are made. It says polypeptides. Remember, polypeptides is just a fancy name for a protein. Alkalians, biofilm. All right, there's the plant cell. Know the three differences between plant cells and animal cells. What were they? Plant cells have that rigid cell wall made out of cellulose. They have this large, giant vacuole in the middle called the central vacuum, which is just a dumping ground, but it also gives turgor pressure to keep the cell in its shape so they can be stacked up and the plant can grow up. And they have these chloroplasts, which contains chlorophyll, which makes sugars through photosynthesis. The animal cell does not, this is the nucleus here, it's not a central vacuole. It doesn't have any chloroplasts. And it doesn't have a cell wall, it just has a plasma membrane around it. So know those three differences. All right, you need to know these organelles, okay? Know what the rough ER is. Why is it rough? It has ribosomes where proteins are made. Know what the smooth ER is. It's a membrane system. All this is still attached to the nucleus, but the smooth ER is where lipids are made. It breaks down carbohydrates and fats, but mainly lipid production. Now, remember what the Golgi body was? That's the UPS store of the cell, where it takes the proteins and the lipids manufactured here and it packages them, addresses them, puts them on the motor proteins, and they go across the microtubules all over the cell. They're delivered. Know what a lysosome is? This here is mitochondria. Know what a mitochondria is? Remember, it's the powerhouse of the cell. The nucleus, okay. There's the rough ER, the smooth ER, good definitions of those. Here's our lysosome. Remember, I'm not going to ask you on the test perioxomes. So there's the central vacuole in plants. The Golgi body, remember that. UPS store, packages, ships. All right, and the mitochondria, there it is. It makes ATP. We're going to learn about that in the next test, how the mitochondria makes ATP. Know what a chloroplast is? That's that organelle in plants that has the chlorophyll, hence the name chlora. So it's a plastid that has chloroplast, absorbs sunlight to make sugar. So know what a chloroplast is? Great thing to know. Know the basic definition of a microtubule and a microfilament. I'm not going to ask you the intermediate filaments. There's our little motor protein. Know the difference between a cilia and a flagella. That's a good thing to know. All right. This is good just because it's going to be repeated so often. What's a cuticle? It's a type of extracellular matrix. It's a waxy layer that's on the outside of the leaves. Know the basics of cell junctions, tight junction and area junctions, gap junctions, know what those three things do. And that's it. So that, we do have a test. Again, I am sorry we keep pushing this thing back. Once we get the first test out of the way though, my prayer is everything's gonna run smoothly from that point on. So, but once we get this first test done, Friday, you have to log on between 10 o'clock in the morning and 11 o'clock at night. Once you log on, you got 90 minutes. Remember, Proctor Track is filming you, so make sure you're clothed in your home. Make sure, you know, if you're embarrassed by messes, you can clean up there behind you. You gotta have that mirror behind you. You have to scan your face, your ID card. Every time you log on Proctor Track, you gotta do that. It's going to watch your eye movement. Just don't cheat, please. It catches people cheating about 95% of the time. It tracks your eye movement, and if you shift your eyes to a certain area and you keep it off the screen for a while, just for a few seconds, it's going to flag it. And so, and then it's going to try to determine why you're doing that. Proctor Track has caught people with their phones down low, so they received Fs. It's caught people with phones trying to take a picture of the screen so they can pass that test information on to somebody else. Don't do that. Just study, sit down, take the test. Don't, you know, again, I'm going to curve this. I tried to make it as easy as I can, but this is biology. It's going to be hard for some people. I know that. Especially, it seems like if you're good at math, you struggle at biology. And, you know, I'm decent at biology, I struggle at math. So there you go. So study hard. There's a lot of information on this test. The next test is more focused. The third test, a little bit more scattered again. And then I'm going to give you some warnings as we go. The last test this semester is by far the easiest. Everybody does great on that last one because it's big things that we know about. So study, and Friday between 10 in the morning, 11 at night, once you log on a Proctor Track, you get 90 minutes. You can't take breaks. So before the test starts, restroom, eat, do whatever you need to do. Get settled so that you can take this test. All right, so study Friday. Then on our schedule, test number 2 is supposed to take place on Monday. I'm not going to do that to you. We'll probably shove it back to Wednesday. The third test we'll probably shove Monday or Tuesday. We'll get back on schedule after the second test. But there's been a lot of difficulties, glitches. This is the last semester and this semester is the first time we're using Proctor Track because of COVID-19. So just bear with us. We'll get there together. And I'm going to work with you. If something happens and Proctor Track just doesn't work for you, don't worry. I will arrange for me and you just to get together on Zoom or Google Meet or Skype or something so that I can just give you the test live. If we've got to get to that point, we'll do it. We're going to get you through this class, hopefully get you through passing, hopefully get you through it for a really good grade. And if not, you know, if we keep running into difficulties, we're just going to see our way through them. So study, we'll see you soon.